Hello, my name is Ruby Sprehe, and I will be presenting my project on an anti-mesothelin recombinant immunotoxin based on Deoxyribonuclease-I. This project was done during the Bridges to Back Summer Research Program. What is a recombinant immunotoxin? What is Deoxyribonuclease-I? And how does this relate to cancer? RITs are made up of a toxin attached to an antibody that binds to a targeted cell. Basically, take the variable fragment portion of the antibody. This variable fragment portion is used to target the specific cells that we want to kill. The toxin is then used and released to kill the cell. One toxin under study is Pseudomonas exotoxin A. Although PE poses to be an effective cancer treatment, its bacterial origin causes an immune response. This causes the toxin to be deemed foreign to the body. With Deoxyribonuclease-I, this does not occur. DNase-I is an endogenous human protein. DNase-I is an enzyme that cleaves DNA by breaking the phosphodiester bonds. Using DNase-I, we predict the immune response will be less likely. Therefore, the immune system will be less likely to mount a response to this drug. Linking this protein to an antibody, the enzyme can target the DNA of tumor cells, causing them to die. This is how it relates to cancer. Here, this is the digestion and ligation of creating the mutant plasmid. We used the SS1 LRGGS plasmid, DNase-I plasmid, and created the mutant plasmid, transforming it into E. coli NEB5 alpha cells. Performed sequencing, autoinduction with BL21 cells. An inclusion body preparation was performed, protein assay, refolding, dialysis, chromatography. We used an anion exchange chromatography, then cytotoxicity assay to determine how effective our RIT is. This is an agarose gel electrophoresis, which determines which samples took up the predicted mutant plasmid. Samples 8 and sample 9 were used for sequencing. This is because the predicted mutant plasmid should have bands at 3.653 kilodaltons and 974 base pairs. Samples 8 and sample 9 closely resemble that match. Sample 9 does show a very faint band, which could be concerning, so we proceeded on with caution. Here, this is the expected plasmid sequence, and we compared the results between samples 8 and sample 9. With sample 9, there is a lot of red. This means that this was not a close match to the expected plasmid sequence. Perhaps the band at the top here is the reason why. Sample number 8 has most correct except an insertion of an A. We resequenced and made sure that this was not gonna cause any issues. We proceeded on with caution. Here is an SCS page. This SCS page shows sample number 8. The predicted molecular weight is 45 kilodaltons, expressed with this arrow. We can see that overexpression is visible at 45 kilodaltons. This means sample 8 was used and predicts that we have our protein. Looking back at the methods, we start with an inclusion body preparation, continued protein assay, refolding, dialysis, and anion exchange chromatography. Here, the results show inclusion body prep, overexpression is visible. This means that our protein was there. Refolding conditions allowed for the protein to be visible, showing that the protein is still there. Dialysis buffer, there is no band. This means that the protein did not leak into the dialysis buffer. We continued on with the load sample. The load sample contains our protein. This is what we will be loading into the column for anion exchange chromatography. However, with the flow-through, there is a band visible. This means that the discarded flow-through contains our protein. This was not what we predicted, as the protein did not stick to the column during anion exchange chromatography. Therefore, purification failed at anion exchange chromatography. In conclusion, we successfully created SS1 DNase-I plasmid, and we've transformed it into E. coli. We performed an agarose gel electrophoresis to determine which samples we wanted to sequence. Sequencing determined which samples to continue with and proceed on with the project. Protein expression matched expected results. This was seen with the SDS page. Purification failed at anion exchange chromatography. This was seen in the last SDS page, as the flow-through contained our protein. Future directions. We want to repeat and modify the experiment to improve the results. Perhaps altering refolding conditions or changing the linker length between the variable fragment and DNase-I will allow the anion exchange chromatography purification step to continue and allow for cytotoxicity assay to be performed. I would like to thank Towson Bridges to Back Program and CCBC for the opportunity, Dr. Lang, Dr. Gut Snyder, and Dr. Berlin Trudy May for the guidance, the NIH Bridges Grant for funding, and special thanks to Dr. Weldon, Jillian Baker, and all the students that have helped with the project. Below are my sources. This concludes my presentation on an anti-mesothelin recombinant immunotoxin based on Deoxyribonuclease-I. Thank you.